In pharmaceutical research, new drugs are first tested on animals. Any predicted effects on the human organism are initially solely based on animal experiments. However, it's not always possible to transfer the results to humans. Tissue engineering offers an alternative. We are guests at the vasQlab at the Karlsruhe Institute of Technology. Professor Ute Schepers explains the process of testing active pharmaceutical ingredients using human tissue without putting humans in danger.
Tissue engineering
Tissue engineering refers to the reconstruction and emulation of healthy or diseased tissue, which is something you typically only find in nature. These days, we can reproduce and imitate this process with various methods. It's possible to recreate organs and use them as so-called organ chips to develop active substances. The vasQ chip is an organ-on-a-chip technology that can simulate several organs on one chip.
Unser vasQlab-Chip besteht eigentlich aus einem künstlichen Blutgefäßsystem, was wir hergestellt haben aus einer...
Our vasQ chip is an artificial vasculature, which we made using a thin plastic film. This vascular system has pores, allowing us to use tissue engineering and apply organs onto the chip or create 3D structures. We can emulate the liver, skin, heart and kidney or brain and kidney and connect them to a circulatory system. This gives you a multi-organ chip to test for toxicity or the effects of drugs. In a manner of speaking, you combine multiple organs based on the effects you want to achieve.
Vielleicht auch nachschauen von Medikamenten, je nachdem, welche Wirkung man erzielen möchte, so packt man die verschiedenen Organe zusammen sozusagen.
The liver plays a very important role in drug testing. It's used to test drug toxicity. A new anti-cancer drug can be tested using a recreated liver to see if it's well tolerated in the human body and if further research is warranted. The vasQlab's mini organs have one crucial advantage.
Wir machen unsere Organchips mit menschlichem Material, das heißt aus menschlichen Zellen.
We use human cell material to make our organ chips. We're well aware that we would have difficulties finding test subjects to test a new drug. We know many clinical trials that have failed because the test persons were subjected to severe harm. That's why it's difficult to find people who are willing to sign up during early phase clinical trials.
Menschen dafür zu begeistern, eben neue Wirkstoffe auszuprobieren.
Organ chips make it possible to determine the efficacy of an active substance or medication in an early stage using human tissue. That's why this method could eventually also replace animal testing.
Das ist genau das größte Ziel eigentlich dabei. In Deutschland sind 3 Millionen Tierversuche pro Jahr.
That's our ultimate goal. Germany uses 3 million animals for testing each year. Many of them are used in modern studies of substances. Needless to say, we could significantly reduce animal testing if we can switch to human material at an early stage. Having said that, we still have a minor problem because legislators require a large amount of animal testing and we must comply. However, you can shorten preclinical development and reduce animal testing in this setting. That's also our goal.
Und das ist auch unser Ziel.
But
Man hat ja nicht den Menschen auf
we are unable to recreate the entire human body on a chip and have a complete organism. Many chips don't include an immune system, nerves or blood vessels, which differ in human bodies. We would have to emulate this as well. It will be a while before we're able to replicate the complexity of the human body. But things are off to a great start. The use of human material in experiments is especially interesting for personalized medicine. Here is one possible application. Let's say you have a tumor model where one patient responds well to a chemotherapeutic agent and has an excellent survival rate. Meanwhile, despite the treatment, the other patient dies fairly early on due to a different genetic background. We can take cells from the candidates and replicate the organs or organ portions of the individual patient. We can subsequently test various chemotherapeutic agents to determine the best chemotherapy for the respective patient. This would give us an initial prognosis of how the drug will affect the patient. And that's the first step towards personalized medicine. According to Schepers, this area has great potential. Many companies around the world already focus on recreating human tissue.
Aber wir stehen noch am Anfang. Wir stehen noch am Anfang.
We're still in the early stages of qualification and validation studies of chip organs as replacements in pharmaceutical research. It will also take several years before we have complex organs to definitively say that the brain, kidney or liver on a chip reacts exactly the way the kidney acts inside a human body. We still have a long way to go before that's the case. However, this area has great potential because everything we are currently developing will be crucial to recreate entire organs as organ replacement systems at a later point. Of course, the big vision is that we are eventually able to produce a donor kidney by using the patient's own human material.